Today's primer will be on the analysis of rare variation. We'll talk a lot about sequencing, some of the kinds of analytic issues around it, and a kind of pointer to some of the emerging work and results that are really starting to become a reality now that we can actually sequence sufficiently large numbers of individuals to take a look at this particular class of variation. Now, prior to sort of thinking about sequencing and rare variation and all that kind of stuff, a very important piece of work was spearheaded by our fearless leader, Eric, really trying to take a kind of comprehensive view to how we think about population genetics, how we think about rare variation, and what that means with respect to studies of phenotypic variation in the population from a genetic point of view. And this paper in PNAS that Orzook led, and I collaborated with Eric and Neale and many others, as you can see up there, really goes through a lot of the kind of design considerations about how we think about the impact of demography and population genetics forces on rare variation, and how that has implications for the way that we think about actually, kind of designing and executing these kinds of studies. Okay, so the, fundamental goal that we have, at least from a genetics point of view, is really understanding the genomic basis of disease. And there are two, main study designs that we've used over the course of the last 10 to 15 years now. One on common variant associations studies, kind of GWAS, although Eric might prefer to style it as CVOS, since, rare variation studies that are whole genome sequencing are also genome-wide, so maybe we should split it based on the frequency of the variation we're looking at rather than the part of the genome that we're looking at. And then this rare variant association study. And the important distinction between these two kinds of approaches really turns around the kinds of analytic and statistical considerations that you have when you're running your association studies. So in a common variant association study, it's pretty straightforward. You have a variant. That variant's pretty common in the population. It's common enough in the population to test directly. That's an awful lot of GWAS is simply a sort of marginal association test of a single variant, contrasting cases and controls, or looking for mean differences in some quantitative trait contingent on what allele you happen to have at that position. And we can contrast that with RVAS, where we aren't in a position where we can actually directly test each of these variants because of the frequency, because surprisingly enough, rare variation is rare, and when something is very rare, it's very difficult to kind of correlate it with something else in the population. And so that means we have to kind of start to reorient the way we think about actually analyzing this kind of variation. So most rare variation analysis, and there's probably one primary exception in terms of the analysis of Denovo variation that I'll touch on briefly, is this kind of approach of aggregating rare variants in a region, maybe a gene. Maybe that's the most natural way of thinking about orienting our analysis of rare variation. And then when we perform that, what we're going to do is we're going to group together these variants, and then we're going to test them for association in a similar fashion as to the way in which we test, say, rare variation. Hey, Lou. It's good to see you. Pretty good. So this kind of rare variation analysis where you're picking a gene, grouping rare variants explicitly, and then proceeding to analyze them in, say, cases versus controls, spawns a whole bunch of additional questions, like what rare variants do you group together? Maybe you want to impose some different weights on those rare variants. Maybe you want to use a frequency filter of different descriptions. Maybe you want to consider some other biological or functional information. The space gets much broader and vaster because it's not this sort of, very straightforward, almost, simpleton sort of approach of how you might perform a common variant association study. Now, again, our kind of fundamental approach is to look for regions where we're seeing a difference in the frequency of a class of variation in cases versus controls. That's going to be the same engine of information that we use for basically the entirety of association studies. And so it's very similar to that, but there are some nuances. All right, so lots of questions. Natural question one is what variance? You can think about loss of function mutations or missense mutations. You know, we have to start to think about what the biological consequences of genetic variation are likely to be in order to design a more careful model. You can ask questions about what frequency threshold you should use, and the goals for frequency threshold, you know, imposition is to really try and maximize the evidence for association at a given, you know, region, right? You want to have the best power possible. And, you know, when you're kind of maybe going up the allele frequency spectrum, that might be doing a couple of different things. It might be watering down the effect size, because maybe slightly more common rare variation has a different sort of functional impact or different risk than much rarer variation. We might have a mixture of effect sizes. And then there are even further complexities that turn around things like, you know, maybe once you get above a certain frequency, all variation is equally likely to be risk or protective, but below you might start to bias. Like that's another reality, and that has implications for how you think about doing these kinds of rare variant association studies. There are sample size questions. I'll tell you the punchline is how big does it have to be? It has to be extremely large. I think I'll give some descriptions about some of the ongoing work currently in terms of that view that we're seeing in terms of when things start to actually yield and appear and those kinds of things. But the sample size guidance from common variant association studies is sort of markedly lower than the guidance necessary for rare variant association studies. So if your phenotype of interest took 5,000 cases and 5,000 controls to start really delivering the goods in common variant association, you're going to need probably 3, 4, maybe 5 times as much of that. All right, so there are some possible upsides and other clever considerations that we can do from a design point of view that can maybe load the dice. In particular, things like population isolates or de novo variation offer some unique advantages in this kind of space that are perhaps distinct from the common variant landscape where, you know, doing a GWAS in, say, Finland is very, very similar to doing a GWAS in, say, America. Like, it's not going to be that different in terms of your ability to detect regions of association. And then there's this question of genomes, and that poses a very broad space of issues around what do we do with respect to non-coding variation. Okay, so the approach to think about a lot of these questions is to just simply build a model. Now this model has some conveniences to make it a little bit more interpretable, and it has some conveniences in terms of being able to really make the kinds of analyses that we do a little bit more straightforward from a power calculation point of view. But I think the model is at least instructive to what I would describe as the first order set of questions. All right, so let's think about now, with respect to rare variation, this idea of having basically 2 classes of rare variation. We're going to have a class of rare variation that confers risk, and we're going to have a class of rare variation that is neutral, okay? And for all intents and purposes, we're going to assume in the context of this model that all rare variant effects are lined up in the same direction, contingent on being risk. That is an assumption. That is probably an unrealistic assumption for some genes. I think for the analysis of loss of function variation, this is a much safer assumption, although you can even imagine circumstances where you have nonsense-mediated decay for some loss of function variation, and then you have other, you know, premature termination of the kind of final, you have your LOF past the 50 base pair terminal exon boundary, and that leads to a sort of malformed transcript that actually does get translated, right? So it could be the case that loss of function doesn't act uniformly, but nevertheless, we'll make that assumption. And so we can think about this sort of typical burden test and break it into a couple of different considerations. So the first key question is the kind of 1,500 base pair coding region. So your typical gene has about that much coding territory. And we're going to do a lot of description of what the typical gene is, and that does not reflect the variability in gene size, the variability in mutation rate that comes with the variability in gene size, but nevertheless can be instructed for like John Q. public gene. And then this mutation class consideration, we have these neutral variations, so they confer no risk and there's no selection against the neutral variation. It's just neutral and it's hanging around in the population. And then there are these null alleles, and they have some increase in risk that we'll term lambda here, and they have some selection coefficient that we'll term S. Okay, so this is the general idea. And the real trick here is that we can specify, based on coding variation, kind of, in a sense, three main groups. So there are silent variants, or synonymous mutations. These are not changing the amino acid at the position, and there's basically no evidence that they're really going to do much of anything biologically in the context of the human system. They're all neutral. Then there are these disruptive mutations, and we can think about those as the point LOFs, the frame-shifting insertion deletions, those essential splice site variants, and they're actually disrupting the transcription of the, they're actually disrupting the transcript, and so you're kind of knocking down the expression of that gene. It may not necessarily be a 50% knockdown if you have some feedback mechanisms and those sorts of things. And then we can think about missense mutations that fit into this kind of mixture class, where we'll say there's some rate alpha, and that rate alpha is the fraction of missense mutations that are going to behave like putative loss of function mutations in the context of the gene. This is obviously a simplification. It's ignoring the idea that there are hypomorphs for missense mutations. But again, it's a model. All models are wrong, but I think this one is instructive. Now, if we say that a typical gene has an alpha of about 25%, and that number arises from doing things like comparative genomics analysis and looking at how many amino acids are fixed different between different species and those kinds of things, and the relative attrition across mammalian species, that gives you some baseline numbers that the number of missense disruptives to null disruptives in the general population is about 2 to 1. So that is to say there's like twice as much mutational target for missense badness as there is for LOF. And then that, the kind of all null to disruptives is about 3 to 1. Okay. All right, so the most important figure to think about, and this really is the single most important figure, is the combined allele frequency. Okay, and what the combined allele frequency is doing is it's representing the sum total of the rate of this class of variation. It's the kind of frequency of disruptive mutations in a gene, combined. And that's really, if we knew all of the bad mutations and we could specify exactly what the biological consequences of each missense mutation were and what the biological consequences of every putative loss of function mutation were, then we could get to this kind of optimum value for the combined allele frequency, right? Because that's going to be like the rate of these bad kinds of variants in a gene. Now, the combined allele frequency has a kind of two-regime model contingent on the degree of selection pressure against that class of variation. So in the event that a site is neutral, then or a gene variation in a gene is neutral, then the combined allele frequency is approximately 4 times mu times NE, where NE is the effective population size, or about 10,000. When selection starts to kick in, and there's actually selective pressure against the kind of genetic variation, the frequency flips to a different regime. No longer under this sort of drift regime, it flips to this regime where the frequency is held artificially lower, and a good approximation for that is mu over S. And to think about what mu over S is meaning here, so mu is the mutation rate, and I want you to imagine for a moment the class of genetic variation that arises but renders you completely sterile. Okay, so that is to say you have no children contingent on having one of those mutations. If you have such a mutation, then we would expect the frequency of that class of mutation in general in the population simply to be the mutation rate, because your parents didn't have that variant because they're not sterile, they had you. Way to go. Way to go, parents. Bringing it home. In contrast, you are now sterile and you can't have any children. And as a result of being unable to have any children, the frequency of that variant is just basically fixed at that mutation rate. Alternately, and so that would be like an S equal to 1. Now, alternately, if S is equal to, say, 0.1, which is actually really, really substantial in population genetics terms, that means that there's kind of like a 10% chance, you're going to have a 0.9 sort of fertility ratio on average. There's a little bit of, drift and, some additional terms swept under the rug, but basically if you have an S of 0.1, the expectation is that that variant will hang around for about 10 generations and then extinguish. And that's the kind of another way of thinking about S when S is large. And, kind of 10 to the minus 3, 10 to the minus 4 S's are the sort of transition point for the two regimes. So this is under what we would call an equilibrium model. Okay, so in population genetics, there's this like idealized equilibrium model. You have a population, they're all stable, they all randomly mate, everything's fantastic. There are no weird assortative mating or other violations of what's going on in the context of the population genetics landscape. And in that circumstance, this is what you get. Well, what happens for the combined allele frequency for other populations? Because humans have not a perfectly equilibrium demographic history. We're an expanding population currently. We've had multiple different bottlenecks. There's been population isolates. There are lots of different kinds of human populations out there from a population genetics point of view. And the punchline is that it's essentially unchanged for the expectation of the combined allele frequency. And so here's one of a series of very complicated pictures. I'm going to try and walk you through it as clearly as possible. This is really like a challenge. All right, so we got the selection coefficient here on the x-axis, right? And that goes from 10 to the minus 6 all the way to 10 to the minus 1. So this kind of 10 to the minus 1 here, that's very, very strong selection. For 10 to the minus 6 over here, that's pretty much effectively neutrality. And then on the y-axis here, we have the combined allele frequency. And what Steve Schaffner in particular ran was a bunch of different population genetic simulations, either assuming equilibrium with 10,000 or trying to do like pretend Finland or pretend Iceland as far as we can tell based on the demographic history of those things, or kind of pretend Europe, or one flavor of expansion where the population is just kind of expanding, or a second flavor of expansion where it's kind of going to go through a bit of a bottleneck and then go through an expansion and then a subsequent massive expansion. You can imagine lots of different models, and pretty much regardless of the model, you can see that the combined allele frequency is basically identical, right? Those lines are all pretty much smack dab on top of one another. Okay, so that's saying that on average, pretty much regardless of demographic history, the expected frequency of a class of bad variation is equivalent across populations. Now, let's talk about individual allele frequencies. Okay, so instead of thinking now about the sum total of nasty bad mutations in a given gene, what we're going to try and do now is think about the individual sites rather than the composite frequency. And you can think about this in two ways. You can think about it as the individual disruptive mutations in a given gene, each of which has a mutation rate, or alternately, you can actually think about it at a gene level. And for both circumstances, it works pretty well, especially when the degree of selective pressure is largely the same against that class of variation. So here's the distribution of allele frequencies. So this is a kind of proportion of alleles below the frequency threshold. That's on the y-axis. And then on the x-axis, we have so here's our x-axis. Here's the derived allele frequencies. That's the new mutation that's arising in the population. And here, these sort of almost parallel lines are different selection coefficients, okay? So what that's saying is that under, neutrality over here, this is what our distribution of allele frequencies would be expected to see in the population. This is, so that is to say that here at 0.1, so at 10%, that's about 10% of our alleles will be below that allele frequency under neutrality and equilibrium, and everything is nice and fancy because that's the way kind of population genetics works out if you make all of these nice assumptions about equilibrium. You can contrast that with things under stronger selection, where here we've got our the topmost line here is this S of 0.1. So this is really strong selection, really bad news. And if you look at the derived allele frequency of 0.001, 10 to the minus 3, then basically you won't expect to see any mutations of that class with this really super strong selective pressure. Okay, does that make sense? You can kind of stare at these figures at the end of the Arvas paper if you want. All right, so let's talk about reasonable ranges for S, where we might expect our kind of population genetics, our kind of statistical genetics analysis to start working. And it's really for S's greater than 10 to the minus 2.5. Half the alleles are basically rare, or less than a tenth of a percent. If we contrast it with Europe, we now see very different shapes, right? Okay, so the neutral stuff looks basically the same. That's not really having any kind of impact on what's going on. But you can see that the distribution of the individual allele frequencies is actually warped. And this is a kind of general phenomenon that if you want, the kind of easiest way to think about this is imagine that a population goes through a bottleneck. Okay, so we've got this effective population size of about 10,000. Then if we have 100 people break off and go and found a new population that they will call Finland, for example. And if they have this 100 people break off and found this new population called Finland, then what's happened is that if you think about the individual bad alleles that they carry, they suddenly get jacked up in frequency, right? So if you've got 100 founders, the rarest any individual variant can possibly be is 1 in 200. And that's way far away from what can happen when you actually have this kind of equilibrium population and things are operating. And so that really drags up the allele frequency of these bad alleles. But at the same time, there are a whole bunch of rare variants that are very nasty that none of those 100 people had. And so they all get basically discarded away from the population. And so they've been now rinsed from the population. And so what you have is a change in the variability in the distribution of individual allele frequencies contingent on S. Okay. So for Arvas, the relevant range, at least in European populations, is S's around 0.1 to 0.001. That's a pretty good, healthy slice of the universe in terms of selection. So how do we think about S from a disease point of view, right? Because there's probably some theoretical link between the pressure of selection and the, range of effect sizes. Remember, natural selection isn't working on an individual variant, it's working on the phenotype. So it's not like, oh, you have this variant, you're not allowed to have any kids. That's not like the way that it happens. It's basically, you have this variant, that gives you this phenotype. Contingent on you having this phenotype, that has an impact on the relative fecundity that you might have. So when we think about schizophrenia, something that I like to work on, prevalence around 1%, maybe reduction in fitness of around 50%, difficult to say historically. Evolutionary psychology is very difficult to prove, I think, in a lot of ways, for example. That kind of translates to an S of 10 to the minus 1.7 if you have a kind of relative risk of 10. Alternately, you can think about something like diabetes, which is much more common in the population, but as a later onset phenotype confers far less in the way of reproductive disadvantage. And that might yield some kind of S of 10 to the minus 2.5 for something that has a threefold increase in risk. All right, so that's like S. So let's keep it simple from the actual practicalities of running these kinds of associations. Let's just focus on these putative loss of function mutations. They're very easy to interpret. It's all, all kind of, the simplest of the simple when it comes to rare variant association analysis. And here's what happens with respect to power. So I think you can look at the picture. That's all pretty, but the table is a lot easier to interpret in terms of a very concrete way. And so the main question that is still, in my mind, somewhat of an open question is, what is the distribution of effect sizes for putative loss of function variation going to look like for the given phenotypes that we're looking at, right? Because now the reason that we like putative loss of function is, kind of twofold. One, they all go in the same direction, and so that means our, analysis can be reasonably well powered. But the second thing is that if they're they're very easy to interpret from a gene point of view, right? So a loss of function mutation in a gene, you're knocking down the expression, and so there's this nice interpretability, upside and bonus that you get over and above. And what you can see is that, depending on where you are with lambda and where you are with, the force of natural selection on that class of variation, you have markedly different sample size expectations. Now, odds ratios of 20 are difficult to come by for most of the complex traits that we're happening to look at. Let's move this up a little bit. Whereas, things that are perhaps more reasonable are more in the 10 to 5 to maybe even 3 or 2 sort of range for most of the phenotypes that we might be interested in analyzing from a complex trait point of view. Okay. LS are great, but remember, when we set up our model, we set up the fact that, a quarter of missense sites are going to be, effectively equivalent to the LOF in terms of functional consequences. And if you have that as a conceptual basis, well, then that means that if you can recognize that class of missense variation, if you can, run like all of the different prediction algorithms and try and concentrate on a class of missense mutations that we think, kind of in silico might actually be deleterious, then that could boost our power, right? Because they're going to have the same effect size, relatively speaking, if they're acting in the same sort of way. And so we'd want to incorporate them into our kind of gene burden testing the frequency of these kinds of rare variants in cases versus controls sort of study. And here's some very funny pictures. And what the funny pictures are showing is like just using the allele frequency threshold, right? Because we've got this two-class mixture. If you use the allele frequency threshold, you can get an increase in power for an allele frequency threshold for missense mutations if you're, draw the line at the right place. But you can actually, sort of lose power compared to the optimal allele frequency threshold, depending on that kind of crossover point between risk increasing and risk, and neutral variation. And so frequency can kind of get you part of the way there. If you use an optimal frequency threshold, you can increase your power twofold. What that optimal allele frequency threshold is unclear, but I'll talk a little bit about how we use that and how we actually use prediction algorithms, which is the other way to try and take a look at this particular problem a little bit later in the talk. But same deal, if you can be smart about prediction, you can again boost the amount of deleterious variation in your burden analysis, and by boosting the amount of deleterious variation in your burden analysis, you have the opportunity of boosting your power to detect association to the gene, which is what I think we're all interested in at the end of the day. Okay, what about population isolates? I made some reference to this kind of, the way that we have the demographic history of a given population changes the way the distribution of individual allele frequency looks. Well, population isolates are like the real bingo scenario here. So that is to say, we have 4 different pictures here. This is a kind of difficult graph to internalize, but what we have on the kind of x-axis is the relative ratio. It's the ratio of the frequency of the class of mutation, so the kind of bad allele frequency in the population, versus divided by the expectation. So everything is scaled to expectation here in the same way. So remember, we have a different expectation for variation that has a mu of that has an S of 0.1 versus an S of 0.01, right? Mu over S will change, so the S of 0.01 will tend to have 10 times as common variance as the S of 0.1 in that mu over S sort of conceptual regime. And so what's plotted on the x-axis is that frequency divided by the expectation. And then on the y-axis, you have the kind of relative number of sites, the frequency of that class of variation, so just how common they are. And what I want you to take a look at in particular is this plot down here, which is representative of Iceland, and this plot down here, which is representative of Finland. And whoops. And what you can see is that as you move the S at different values, contingent on demographic history, you're going to stretch out the distribution of the frequency of an individual variant over the expected frequency. So that is to say that there are things in Finland in the S of sort of 10 to the minus 2 sort of range, this sort of red line here, that are maybe 10 times as common as they should be because of that demographic history. Now, what's really great about that is that it's very easy to pick off those genes. If something is 10 times as common, that's like having 10 times as much sample size, which is like a real big bingo in the scheme of performing these kinds of analyses. And so that's what population isolates really buy you in the context of rare variant association study designs, is this idea that for a subset of genes influencing a given trait, the frequency of those bad mutations in those genes is far, far greater than you would expect to see based on the demographic history of, say, Europeans. And as a consequence of that rapid, that dramatic increase in frequency, we have much better power to detect. But it doesn't mean that we're going to capture all of them, right? Because where there are winners in the let's increase in frequency above our selection coefficient would indicate, there are also losers. There are things that are absent from those populations that are not going to be observed because they weren't there at the founding of the population. They didn't exist when the bottleneck happened. Does that make sense? Are we like following this kind of, I don't know, population genetics at 8:30 in the morning is like, heavy going, man. It's really heavy going. All right. So one final piece, but you've made it through most of the population genetics. So count yourself lucky. You've had like that stiff dose of population genetics from 8:30 to 9 o'clock in the morning. And now we're going to talk about statistics. So much easier. So one other thing to think about and just remember when you're performing these kinds of rare variant associations is that even when you are grouping rare variants together, they're still rare. And as a result, you may not have so many copies of the allele in your overall data set. And with that, you can end up getting some problems in dimensionality. So what we have here are two QQ plots. And the QQ plots are showing our kind of theoretical quantiles for the, say, the chi-square of 1 degree of freedom for the p-value, and then the kind of observed simulation of what we get out from doing different tests. And I want you to notice that the tests in general are sort of below the line. They're all deflated, okay? And this is in a scenario where we have 40 copies of the minor allele in, say, 2,000 individuals. And then over here on the right, we've got the scenario where we've got 80 copies of the minor allele in, say, 10,000 individuals. And there are a couple of really important take-home messages. The first is 80 copies is about the transition point where most statistics, say the allelic chi-square or the Armitage trend test, become, relatively speaking, well-distributed. That is to say, they become well-distributed up to p-values around sort of 10 to the minus 8, give or take. And that's an important line because if you're well-distributed up to 10 to the minus 8, then you're actually straightforwardly testable under the null. If you have fewer than 80 copies of a variant, then you are in low kind of dimensionality space, and the actual kind of statistical association tests that you can perform under the null tend to be quite deflated. We don't see this full range of p-values that we would expect to see by chance. And that really, in a sense, caps some of the way that you can perform these kinds of analyses. Now this holds for equally balanced case versus control analyses. It doesn't, it starts to shift around a little bit if you invoke something like a Fisher's exact test, and if you have very large number of controls compared to cases. That can actually alter some of your power to detect association. For almost all of these kinds of studies and tests, the only class of variation that we're really in any way powered to detect is that of risk-conferring variation. The detection of protective variation for a disease outcome, especially one that is rare, like schizophrenia, is markedly weaker. It is, like an order of magnitude more sample size required sort of scenario. It's pretty grim. Okay, so dimensionality, think about the way your test statistics are distributed, because that has an impact on the way in which you actually can, perform your statistical tests. Okay, so you've heard from Geraldine and from a bunch of others about, kind of rare variation. I think Daniel is going to talk to you maybe next week about Nomad and aggregation efforts and those kinds of things. And I just want to highlight the kind of typical slide that you've seen many times already. This, data size is accelerating in the context of sequencing. And, what I wanted to highlight is that, when I started doing this primer on rare variant association studies, this is what the kind of pipeline would look like. So this is a pipeline that my postdoc, Andrea Gana, kind of knocked together to describe basically the QC of exomes or genomes. And back in the dark ages, we would use half a dozen different tools to perform these various operations, and it wouldn't work so well. And it would take a long time to run these kinds of things. Like, this is really a big challenge. Like, just doing QC of a kind of 8,000 exome study, which is still just about typical, I guess, for the kind of case control, would take like a day of compute, would take 2 weeks to actually run, lots of different pipelines and all that kind of stuff. And so one of the things that we've done is kind of start this hale effort. I think many of you have heard about hale already. But the whole model behind hale is that this toolkit, and I'm just going to give you a little advert for it rather than spending too much time, enables us to kind of basically move away from the old model of data analysis and high-performance compute, where you have this big pile of data, and you sort of shovel that data to the different machines, but you try and keep it all together to the best of your ability, to a kind of distributed data model where you split the data up into lots and lots of small individual little files that capture different parts of the genome, perhaps, depending on how you want to slice and dice your data set. And then it's all built on basically the Apache big data stack, right? So tools like Spark and Scala and Hadoop and Parquet, all of these sort of cloud-based computational approaches are being leveraged in order to kind of design this toolkit, hale, to actually cope with the scale of the data. But there are a bunch of mathematicians. Cotton's a mathematician. Alex, John are all mathematicians by training, and so they like to think about algorithmic approaches and kind of doing linear algebra in an effort to expedite the speed and scalability of this activity. You can move this pipeline into like 9 minutes now. So to QC an exome data, it'll take like 9 minutes on the Google Cloud, probably even a little bit less. After the various improvements, people are like shaking their head like, how did that happen? Like, and that's the nice thing about scalable computing, is that you can make it go very fast in real time. You do pay a little bit more. There's a little bit of an extra compute cost because, there's a sort of capital cost of firing up these kinds of instances in the scheme of things. And that hale was what was used to actually do the QC and, kind of define the data set necessary for things like Nomad and Exac, and that can be turned around and optimized. And the other thing that I would comment about these kinds of scalable approaches and the importance of being able to actually get answers in something approaching real time is that you have the opportunity not to do the QC just once, right? For any of those of you in the audience that have actually done QC of a genetic data set, you do it a lot. Like, you do that thing over and over and over again, because you like do some QC and you're like, maybe that works, and then you look at your distribution of test statistics and you're like, well, maybe I'm not so clever, maybe I need to go back and do that again, and you go over and over and over again. And the analysts are like nodding in the audience like, yes, this is my life. Don't worry, it was my life too. Okay, so you can learn more about Hale at hale.is. It's a really useful toolkit for performing these kinds of analyses, but you really do need to like embrace the cloud. You need to kind of move away from our good old days of high performance compute cluster and move towards this cloud scenario. There are people institutionally here that can help you get set up with accounts on the cloud and help with how billing and that kind of stuff can work. I'm sure Lucas isn't going to be happy for me to say this, but Lucas Carlson's been a really good resource for that, and so if you want help, feel free to reach out to him or IT. But there's also like a chat, and there's a couple of different fora that Hale developers hang out on, and they're really eager and enthusiastic about helping to build things forward. Ooh, that is not the slide I want now. All right, let's skip that slide. We'll go here. Okay, so let's talk about Denovo variation. So we got like 15 minutes before MPG, so we got some time to go through some actual results, hopefully. 15 minutes, 15 slides. So over the last 5 years or so, yeah, that's about right, this kind of wave of de novo variation identification and analysis has sort of gripped, in particular, the autism community, but also to a lesser extent some other communities, including, say, epilepsy or some of the kind of cardiac hypertrophies and those sorts of congenital heart disorders. And the model here is to look for newly arising variation. Now, I mentioned when we were going through that like population genetics log that you've probably all whited out in your memory because it was so horrible, that de novo variation offers a kind of unique opportunity. Now, one of the things about the rare variant analysis that again was sort of swept under the rug is that for all of those power calculations, we assumed that we knew exactly what the frequency of all genetic variation was in the general population. So that is to say we had like 10 million controls from every population that has 10 million people and could actually tell you what the expected allele frequency is empirically for basically every gene in the genome. Well, we're not quite there yet. I think we have a little ways to go, but that's maybe a future goal for us to set ourselves. Presents for future us. But what we do have is actually a reasonable handle on what the mutation rate is in the human population. And by having a handle on what the mutation rate is, we can actually build a model to figure out what that expectation is. And if we know what the expected mutation rate is, we can now do a slightly different flavor of test. So rather than testing against the estimated allele frequency in the population based on a very large control sample, we can estimate, we can build a model to determine what our mutation rate ought to be. And by building that model, we can then test directly against that expectation. And that's what we've been doing in conjunction with Caitlin Samoha, who's a PhD student in Mark Daly's group, and Mark, and now Jack Kosmicki and others. And really, this de novo analysis is all built around kind of expectations. And so you can do this either empirically with the control trios that people have done right here, but when you look at what our model predicts for the rate of the kind of LOF mutations in given genes, we get pretty much exactly what we see in the controls, which is somewhat encouraging. And then when we look at different case groups, the DDD, the Deciphering Developmental Disorders project, led by Matt Hurls and Jeff Barrett and many others at the Sanger, that is really engaged in large-scale exome sequencing for patients with developmental delay. They see this strong enrichment of putative loss of function mutations, kind of twofold increase over the background, and then you kind of look at the autism spectrum disorder slice of the universe and you see 1.5-fold increase in the rate of these mutations. And that's not too distinct from what you see for epileptic and encephalopathies. Now, what we can then layer on top, and I think Daniel's going to talk about this a lot next week, so I won't dwell on it too much, is this idea of kind of leveraging patterns of genetic variation from human populations to determine those genes that are under strong purifying selection versus those genes that are, relatively speaking, not under so much in the way of purifying selection. By and large, loss of function mutations are basically selected against for almost every kind of gene in the human genome. We see fewer LOFs than we would expect to based on the mutation rate, in contrast to things like the synonymous variation. But if we kind of try and stratify our genes based on this degree of purifying selection, what we do is we really concentrate the signal in the context of autism. So we move from that relative risk of, say, 1.5 to now a relative risk of 3.25. And in the context of developmental delay, that moves from 2 to 6, so nearly 7. So that really makes a substantive difference in terms of the relative increase in risk. And not only is this doing a good job in terms of concentrating the signal, the stuff hanging about in the genes that are, relatively speaking, tolerant of loss of function mutations, they don't really show any difference in the rate between cases and controls. So at least for this set of disorders, this is where the majority of the strong-acting mutations are. I'm not going to say that there aren't LOS that are hanging out in the rest of the genes that are conferring risk to autism. There assuredly are, but their effect sizes are very, very small. So this is a way of really honing in on the kinds of effect sizes that we'll need to kind of get the job done. Okay, so if you take this apart, then what you see is that for this excess in at least autism, there are a handful of genes. So these genes up there on the right-hand side, they're the kind of tail end of the distribution. These are the genes that are pretty much legitimate lockdown candidates for causing some or conferring some risk to autism spectrum disorders. In almost no case are they what I would describe as fully penetrant mutations for autism spectrum disorders. So I really resist the description of them being causal, which I think is a kind of bandied about a lot. They're little c causal, not big C causal in my mind. And the majority, almost all of these genes in truth, confer risk not only to autism spectrum disorders, but also to developmental delay and intellectual disability. And I think that's kind of one of the emergent challenges with respect to the analysis of rare variation is that when you're looking at a class of genetic variation that is under strong purifying selection, I think the probability that you have like severe pleiotropic consequences is actually markedly increased. So these mutations are not only disrupting autism, they're also like disrupting a lot of, say, cognition, a lot of other communication phenotypes that may be, you know, kind of along for the ride in our definition of autism. If we boost sample in a case control fashion, right? So like de novos are great, but remember, de novo mutations work extremely well as a kind of analysis when you have very strong purifying selection. So S is very big, and when you can get parents. And for a lot of things, we can't really get parents, or for a lot of the collections that are of convenience, they're not, you know, as readily available. And so over the last, say, 3 or 4 years, we've developed a partnership with some folks in Denmark to basically extract DNA from blood spots based on a register that's been held at the State Serum Institute for the last 35 years, 36 years now. May 1st, 1981 was when this process of basically biobanking all of the Guthrie cards for neonatal screening of inborn errors of metabolism kind of happened. And this guy, Mads Hollegard, who passed away a few years ago, pretty much dedicated his entire life to taking blood spot material, making DNA amplification work, and making it available and appropriate for the analysis of, you know, genetic kinds of investigations, including genotyping and exome sequencing. And, okay, I think it thinks that, thank you. It's telling me MPG is starting in 10 minutes. MPG is starting in 10 minutes. So what we've done with exomes with, and it's telling everyone that, now it's decided to wake back up. There you go. You all get to know that MPG is starting in 10 minutes. That's good. It's a 10-minute warning. That's fine. I'm like above my pace that I need to be to get through the remaining slides. So this, you know, exome analysis with the Danish Blood Spot Project, that's Kyle Satterstrom, who's working with Mark and myself to drive forward the analysis of autism, ADHD, and also looking at comorbid patients. And if we have our like de novo mutations, right? So here's our de novo mutation rate in cases versus controls in that class of genes that are loss of function intolerant, focusing on those really nasty, you know, LOFs. Well, our case control rate looks, not too distinct, right? So we see the same signal. That's very encouraging because this is a register-based diagnosis of autism spectrum disorder. So, who knows what was in there? At least that's what some people who are particular phenotype hobbyists believe in. But in truth, what we see is the same sort of increase in rate as when we look at the inherited plus the de novo variation in the Simon simplex and the autism sequencing consortium effort, okay? And the reason that I think this is instructive in a primer context rather than just simply showcasing the science is that it is possible to get there with a case control analysis in the same way that you can get there with a de novo mutational screen. And the case control analysis carries a lot of the same signal that the de novo variation analysis carries, but you do need to have sufficiently large numbers of controls to make those kinds of analyses work appropriately. And that's kind of where the big trade-off is. And then the second thing is, it is worth bearing in mind that for most variation where S is modest, so for S of 10 to the minus 3, almost all of the genetic variation of that class will be inherited in the population. It will not be newly arising. And so the real big kind of deliverable for de novo models and de novo approaches are when the class of variation extinguishes itself very rapidly in the general population. And that's what we see for that kind of top end tail edge of those genes. But as we've started to move further down the list and more into the meat of things, that kind of weakens our signal there. We're seeing not so many obvious candidates arising from the de novo analysis, and actually the case control will start to power it more. And there is that kind of trade-off between that, depending on what the genetic architecture of your phenotype looks like and the degree of selective pressure against the class of variation. Looking at autism and ADHD together, we do start to see a handful of kind of new genes emerging from a case control paradigm in contrast to the kind of de novo mutational analysis. MAP1A is almost certainly a real hit, but we're not quite at the degree of significance that we would be extremely confident about, but this kind of works. But by and large, we see the same kind of enrichment in ADHD as we do in autism. Okay. Now, we can, kind of stretch this analysis considerably further by trying to look at the same class of genetic variation. So what we're going to do is we're going to focus on these putative LOFs, these damaging missense mutations, and we're going to look at them across a wide variety of phenotypic outcomes. And we can do this here at the Broad because people are willing to share data and work together. That's a great thing. We should all be happy to do that. And so the kind of main driver behind this project's a postdoc of mine, Andrea Gana, who's kind of up here. And he's worked with a whole bunch of the other analysts and other scientists to kind of pull this effort off, including Kyle, Miriam, Julio, Jason, Jessica, Micha, and Manny. And so the first thing to look at, again, for these same protein truncating variants, we're focusing now on ultra-rare variation. And what I mean by ultra-rare variation is variation that we don't see basically more than one copy. We don't see any copies of it in something like exact for the samples that aren't overlapping. And when you do this burden analysis, what you see on the kind of y-axis here are the different phenotypes that are very challenging to read. But the top phenotype is intellectual disability, followed by autism, followed by ADHD, followed by schizophrenia, followed by bipolar disorder. So across the kind of neuropsychiatric end of the spectrum, this class of variation that has very strong purifying selection against it seems to carry a general risk. There's this burden, there's this increased frequency of this class of variation in cases in contrast to controls. And that is distinct from what we see for early onset myocardial infarction, for type 2 diabetes, for Crohn's disease, for ulcerative colitis, or more generally, inflammatory bowel disease. So by and large, this class of variation seems to be having a lot more action in neuropsychiatric phenotypes. Now that might have something to do with age of onset. It might have something to do with organ of interest, right? So these are all brain phenotypes, and the brain may be particularly picky about what genetic variation is hanging around in the population, in contrast to, say, your pancreas, which might not be quite so picky, I don't know, in contrast to your lipid homeostasis or some of the other cardiac phenotypes. But we can also look at some continuous phenotypes and ask, again, the same directional question. And the phenotypes that we observe a significant impact of this class of variation on include years of education at the top, age of enrollment in the study. That's a great one, isn't it? Age of enrollment in the study. You know, working on the genetics of aging is a kind of bitter and ugly history in the context of, say, at least common variant association. But, you know, this could reflect premature mortality. It could reflect likelihood of enrollment in a study, right? Remember, when we're talking about age, we got a lot of different things that we can talk about, and that can definitely be one of them. But you know, it's controlled for in each of the various case control analyses. And then the third one off the block is height. So there is actually evidence of what I would describe as directional selection against short stature, at least as it comes with respect to this class of genetic variation. Whether that's pleiotropic, caught in the crossfire, or whether it's, you know, part of the selective pressure, who knows? That's one of the difficult things about these kinds of analyses. It's very difficult to interpret what's happening downstream. Now, I just want to close on something that is important to think about, especially as we've just had, you know, 58 minutes of discussion of rare variant association studies, and that's putting this years of education result into context. So what Andrea did next to try and, you know, really clarify the situation was take each of the different classes of genetic variation that we have seen associated to years of education in the general population, created a normalized score, so all of them are standardized, and then put them, you know, kind of ranked top to bottom in terms of the relative change in years of education as a function of one standard deviation change in that score, okay? So from top to bottom, what we have are the common variant polygenic risk scores is at the top, followed by this damaging and disruptive ultra-rare variation, followed by runs of homozygosity, followed by pathogenic copy number variation, okay? And what you can see is there's like that real, you know, that thing all the way over on the left that's got the strongest impact on years of education is the common variant polygenic risk score, okay? And now remember, this is a polygenic risk score, so it's like full of variability and uncertainty around the effect size. There's a whole bunch of stuff that's unknown about what the actual true effects of each of the various common variants are. But it is representing, at this point in time, the lion's share of the genetic variation that is influencing phenotypic variation, at least as it pertains to educational attainment at this point in time. Meaning that if you are really interested in doing things like epidemiology, and you're really interested in doing things like the characterization of heritability, or potentially even like trying to understand the biology, common variation might be a more important thing to focus on than rare variation is at this point in time. That is not to say that rare variation analysis does not have a role, but its role should be restricted to that which it is good at. And the thing that it is good at is nominating very specific genes, because rare variants in genes can give you a very kind of conclusive answer about whether or not that specific gene is involved in risk for disease. And that is a very useful deliverable, since we often struggle to do that in the context of common variant analysis, right? We get a GWAS, and we don't know what the gene is, we don't know how to link it. You know, we got a lot of problems. We got like 99 problems, and the gene is one, I think, is maybe how I'll conclude. So with that lovely kind of hat tip, I'll conclude and say, you know, scale is possible. Rare variant association study designs are challenging, and they will require considerably larger sample sizes than the sample sizes necessary for common variant analysis. Don't forget common variant analysis. They're like still important. I know sequencing is all the new hotness, but it is important to remember where the signal lives and what we should be trying to focus on as geneticists. But nevertheless, sample size is still king, and with that, I'll close and say thank you.